Hello, I'm Rashad Batley. I'm in the group TEB, and today I'm going to be talking about my investigation of the role of the gene Brady1G25180 in Brachypodium Distachyon. First, the gene structure as well as sequence and protein sequence were determined using online software. It was shown that it had approximately 7,000 base pairs, around 750 amino acids in the chain, and 7 exons, which you can see in the picture on the right. This was corroborated by using express sequence tags in an online library, which helped finalize our working map of the protein, as well, or the working map of the gene as well as its structure. A literature review was conducted to see what the role of the protein might be. As you can see, it has several domains, including a sugar or mannose recognition domain, which is usually involved in immune response, another large domain involved in immune response, as well as 2 domains that are often associated with self-incompatibility. There are some problems with this conclusion, however, since Brachypodium, as well as many of the species shown in the phylogeny, actually can self-fertilize, including Arabidopsis and Oryzis sativa, which, as you can see, is the closest relative in terms of sequence similarity to the gene in question in Brachypodium. So further investigation was carried out to confirm whether or not the online software was correct. It started with investigation of mutant versus wild-type plants. As you can see, there's a severe mutation here at the 3,473rd base pair, which changes a T to a G. This actually supports our conclusion about the gene's structure, because in any other reading frame other than the 3rd reading frame, it would have been relatively inconsequential. But in the 3rd reading frame, it's a non-stop mutation, meaning that ribosomes that read over it, instead of stopping, carry on adding on amino acids, which can lead to an overly long protein that misfolds and can no longer serve its function. These are the primers used for PCR. DNA was extracted and quantified on a gel as well as via nanodrop. And as you can see, it was not entirely successful, which would be a hindrance later on. There was a lot of RNA contamination. The RNase free gel is on the left, while the RNase one is on the right. The lines are not very clear, which is further supported by the nanodrop quantification. As you can see, only mutant 2 really had any meaningful amount of DNA. The rest were much lower. And this would mean that for Sanger sequencing, only mutant 2 would be sent in, which, considering the results of Sanger sequencing, was very frustrating. As you can see on the gel, the sequence falls around 850 base amino acids long, or base pairs, which is the exact region of the gene that was sent in for sequencing. So the primers were effective, and the right sequence was selected. However, as you can see boxed in black here, it was actually a T at the location where we expected a G. This is the location of the mutation. This actually means that the genome that was sent in mimics the wild type version, not the mutant version. The rest of the sequence is very high quality, indicating that the plant gene that was sent in likely came from a wild type plant, which is confusing. It's likely due to human error at some point in the chain, although it's unclear where that human error occurred. And this made it very frustrating that we couldn't send in more plants due to the unsuccessful DNA extraction, because it would have been nice to know if all of them were wild type like this instead of mutant. Phenotype analysis was carried out using histology. These are cross sections of the stems. On the left is a wild type and on the right is a mutant. And as you can see, there's no immediately obvious structural difference between the two. No formal analysis was carried out to determine whether or not the cell walls were different in thickness or anything like that. This could be because perhaps all of the mutants that we have on hand are actually wild type, as suggested by Sanger sequencing, but there's no real way to know, which makes the unsuccessful DNA extraction even more frustrating. But the same conclusion was applicable for the pH HCl staining as well as the T-blue staining. There were no obvious changes in structure, which is honestly not that unexpected, given that this protein likely serves a role in immune response rather than cell wall structure. But it would have been nice to know from Sanger sequencing. Gene expression databases online were used to see where this gene was expressed. I expected it to be expressed in the carpel, or the female reproductive part that pollen is responsible for integrating with. However, it was expressed widely across the plant as a whole through root and stem tissue, as you can see in both the graph on the left and the pictures on the right. The same applies for the orthologous genes in Arabidopsis. It was not expressed in the carpel, rather it was most highly expressed in root and leaf tissue, indicating that it most likely serves some other immune role rather than specifically for pollen. It could be for pathogen detection. It also responds to cold. As you can see, light does not affect it. However, cold does. When a paired t-test was run on various days of cold exposure, all of the p-values were very significant, indicating that cold does have an effect. So abiotic factors may also affect its expression. To test this, an experiment was designed in which salicylic acid would be sprayed on treatment plants, and they would be compared to control plants to see how gene expression of the gene of interest changed. If no change was seen, it would be a good indicator that either pollen or pathogens would elicit an immune response from this protein. But if we did see a change, it would be more likely that something like salt or cold is responsible for, is part of the pathway that this protein is responsible for. RNA was extracted and quantified. As you can see, it was successful this time, but on the gel, ladder was loaded into every well, so that wasn't entirely successful. And finally, for the qPCR results, there was no significant difference between the control and treatment plants, indicating that salicylic acid did not have an effect on expression, indicating that it's likely not involved in an abiotic stress pathway. And finally, this can lead us to conclude that biotic stress is most likely what this protein is responsible for. It is less likely we find that it's responsible for pollen detection, given its expression across the whole plant, and it is most likely involved in pathogen detection. In future experiments, I would like to have a more successful DNA extraction so that Sanger sequencing could be made more effective. And then a formal analysis of histology results would be nice as well to see if it has a function in plant structure. Thank you very much.